All right, so let's keep talking about this viral replication process because I want you guys to think about it and think about ways to block viral replication. There's all sorts of different ways to stop this. Do you stop it at step 1, step 2, step 3, 4, 5, etc.? There's a lot of different ways for us to develop a defense against this. And sometimes you need multiple defenses because the virus is going to keep evolving and changing and working around our defense. So the more defensive strategies we have, the more successful we will be in blocking viral infections. Okay, so we talked about lytic cycle last time. Got to identify and attach to the host, get inside the host cell, take over the host, replicate the genome, build new viruses, and then release them. Bad stuff. So an example of a virus that follows a lytic cycle is something like the flu. It hits you, it knocks you down, it spreads, and then hopefully your immune system gets rid of it. Virulent viruses tend to be fast acting, and you recognize when you have it for the most part. Like, okay, I got the flu, I feel terrible, I have the symptoms. The lysogenic viruses play by a little bit of a different rule, different strategy. So they still have to identify the host. Get out of there. They have to, all right, they have to get inside the host, but then their third step is what's known as integration. So instead of taking over the host, I'm not sure where this is going to fall in. The viral genome blends into the host genome. So their DNA literally becomes blended into your DNA, your host DNA. And now you are going to continue to carry that viral genome. So the fourth step here, oh, that should be replicated. So that viral genome is replicated every time your host cell goes through the cell cycle. These viruses never go away. They're with you your entire life. So they're never going to leave your body. They're constantly part of your new genome. So when we look at things, different viruses, different health issues, what we can do is look at your genome and say, okay, did you pick up a virus at what point in your life? And identify viral infections throughout the course of a person's life. It alters their genome. That potentially can be passed on to their offspring. So some viruses take the lytic cycle, other viruses take the lysogenic cycle path. It just varies from virus to virus. Now, what we'll see with certain viruses is that they're going to be with you your life. So here's a bad example. Herpes, the gift that keeps on giving. Okay, so again, in general, we think of viruses as bad things, but I do want you guys to remember there can be some very positive uses for viruses. Biotechnology, genetic engineering, etc. The virus is simply carrying information. If it's bad information, yeah, it's a problem. What if we switch out the bad information and put the good information in there? So think about genetically engineering somebody who has a genetic disorder, cystic fibrosis. You need to change a gene within their lung tissue. Could we use a virus to insert the correct gene into that lung tissue so then those cells produce the correct protein and you eliminate a genetic disorder that kills people? So Get the knowledge, pull it all in, look at the concepts here, and then I want you to definitely be able to come up with some applications for these things. So lots and lots of possibilities. All right, nothing's off the table. There are no crazy ideas. All could be possible. Jurassic Park could be possible. Use viruses to transmit the information and move genes around. So, all right, so general overview. Here's your lytic cycle. When we're looking at a virus that will attach, well, it's got to identify and attach, penetrate. It goes in, hijacks the host cell, builds or assembles new viruses, and then it releases and it ruptures the cell. So the cells explode. They get destroyed. That's usually how you know you have this virus, because your cells are exploding, you have symptoms. That produces headache, flu-like symptoms, fever, cough. In the case of something like Ebola, bleeding. Internal organs just get mushed and destroyed from this virus. Lysogenic, this is the one where it just kind of blends in and it hides and you don't know that you have it. And then maybe at a certain point it peels off and gets triggered over into the lytic cycle, but then it still stays in you. It doesn't disappear. So that's the crazy thing about the lysogenic viruses. They may go into, we'll call it remission, and you think, oh, it's gone. I don't have it anymore. No, it's still there. It's just waiting for the opportune time to then shift into lytic and go through that cycle. So here's the one I want you guys to think about. Hopefully nobody's had to deal with it, but if you've had chicken pox, writing is terrible here. What issue later in life, as you get older, do you need to worry about? I'm just going to put the big question mark here. Okay? Because that lytic, or that chickenpox virus, is doing the lysogenic cycle. It sits in your DNA and waits and waits and waits and waits, and then later in life, boom, it comes out. Usually when your immune system's down, you're stressed, your body's just beat up, and you're like, oh, I feel terrible. That's when the virus takes advantage. There's environmental triggers. There's all sorts of things that trigger those viruses to go from lysogenic into lytic. You have an expression of it. You beat it back. It's still in you. It can do it again later in life. So again, those can be very, very concerning when we're talking about viral life cycles. Okay, so let's take a look at this HIV, human immunodeficiency virus. In the systematics lecture, we talked about the origin of HIV. This is the virus that leads to or causes AIDS. Evolutionarily, it came from primates. There's a simian immunodeficiency virus, SIV. Simian refers to the apes. And that virus evolved. It jumped the barrier. And now it's infecting, or has been infecting, people. So what we're looking at is the first cases in humans, in the US, I was supposed to say US, date back to about 1981, first identifiable cases. It's believed 1940s. The first human cases. These were in Africa. Oh, let me, so you can see it. All right, so it is possible that this virus has been around longer than that. We just don't know. But confirmed cases of HIV date back to the late 1940s-ish, early 50s in Africa. And it took about 40 years to make its way to the US. So, what's our risk? What's our concern? Well, some people, believe it or not, are actually resistant to HIV. They don't get HIV. They're immune to it. Other people can get infected by HIV and they'll be HIV positive, but they never progress to AIDS. AIDS is the acquired immune deficiency syndrome. That's when your immune system just collapses and you basically die from something like pneumonia, the flu, general bacterial infection. So people who are HIV positive, COVID-19 is a death sentence because their immune system won't be able to fight it off because they realistically don't have much of an immune system. So, again, those people HIV positive but never developed to AIDS, okay, you still have your immune system. And then there's other people, very minimal resistance, and they die from this. They die from HIV. That progresses to AIDS and then general whatever, cold, flu, bacterial infection. Secondary infections is usually what kills somebody who has AIDS, not the AIDS itself. Now, what's crazy here is the resistance aspect. It's thought that the resistance to HIV is due to selective pressure that was caused in our ancestors when they were exposed to smallpox. So if you have the genetics for resistance to HIV, go back through your family history. And did you have a family member who got smallpox, survived it, and then passed on the genetics from generation to generation to generation? Okay, so the key here is this thing called the CCR5 receptor. This is the critical receptor for HIV to get into the cell, to recognize the host cell. So right there, CCR5. So you're like, okay, do I have a CCR5 receptor? Well, if you do, HIV can identify your cell, it can attach to your cell, it can get into your cell, and then you now have, if you've been exposed to HIV, you now have an HIV infection, you're HIV positive, you progress to AIDS, and unfortunately will most likely die. Some people have a modified gene for the CCR5 receptor. So here it is, you're modified for whatever reason. Your CCR5 is tweaked a little bit. So you get infected because HIV still can attach, but it doesn't progress. It doesn't infect the normal route. Something's different. The third choice here, the third option, is people have this really modified CCR5 receptor. HIV cannot attach. It cannot recognize your host cell and actually attach to the host cell and get into the host cell. So here's where I want you to start thinking big picture. Societal benefits. What if we find a way to modify the CCR5 receptor on people? Is that a good thing? Let's think about people who are at high risk. Police officers, first responders, anybody working in healthcare. Would it be wise to modify their CCR5 receptors so they would then be immune to HIV? Just throwing possibilities out there, ideas to discuss and think about. So the way this all works when we talk about HIV is HIV targets these cells called CD4 cells, CD4 plus cells. These have a special group that are called helper T cells. These are basically immune cells. So the HIV goes after those and destroys those, destroys the immune system cells, and this again is what is going to crash your immune system eventually. what's challenging here is clinical symptoms usually appear 8 to 10 years later after the initial infection. So here's a hypothetical to think about. If somebody shows up with these health problems and they are diagnosed as HIV positive, it's supposed to be a V, in 2020, they need to look back over the last 8 to 10 years of their life and go, where did I possibly pick up this HIV? Starting in 2010 all the way through 2020, who have I potentially been in contact with that could have given me HIV? Who in those 10 years did I possibly transmit HIV to as well? So you guys probably have heard of this idea called contact tracing. It's exactly what you have to do here. You have to look back through your history of life and look at who in your life in the last 10 years could possibly have given you this, who could you possibly have given it to, and then start looking at all those people. Do they have HIV? You got it from somebody. Somebody in that chain or in that history has that infection. Who did you possibly give it to? So those are all the things we need to do with contact tracing to figure out how to stop the continued spread of this virus. The challenge is it sits latent for 8 to 10 years usually before clinical symptoms show up. Okay, in our next part of the lecture, I'll take you kind of a brief tour through what HIV does and how it hits the cells, just kind of an overview of the big picture of it, and then we'll talk about treatments, retro or viral treatments that are being used to block HIV and extend the life expectancy of anybody who has HIV.